Hello, everyone, and welcome to the lecture on Chapter 6 called Acellular Pathogens. Before we get started, you may have noticed that this lecture is a little bit on the long side, and so it may be good if you break it up into pieces, maybe two pieces. I will probably note somewhere in the lecture a good stopping point just to take a break, probably around the halfway mark. So I'll probably let you know when that is. So in this lecture, we are going to move from talking about cellular organisms like bacteria, fungi, etc. to acellular pathogens, namely viruses, which are one of the main 6 categories of microorganisms. Acellular pathogens refers to biological entities that are not composed of cells. That's what the word acellular means. It means not composed of cells. They invade and infect hosts in order to reproduce, and for the most part, when we're talking about humans and we're focusing on clinical microbiology, our emphasis falls on viruses, including viroids, virusoids, and prions. And we'll probably talk about these at the end of the lecture, but the main body of this lecture is going to focus on viruses, which are just one type of acellular pathogen. So what is a virus? A virus is an infectious agent that, as we said, is not composed of cells, but rather of a nucleic acid, which can either be DNA or RNA enclosed in a capsid, which is a protein sheath that protects the nucleic acid. And you can see a basic depiction of a virus here on the right hand side. So you've got nucleic acid shown as the purple ribbon, and we have a protein capsid shown in blue surrounding it. Viruses have some unique characteristics in that they are extremely small. Even compared to other types of microscopic organisms, they are very, very tiny. They are on the order of 20 to 1,000 nanometers in size. And in just a second, I'm going to remind you how that size compares to other types of microbes. They are acellular, so they are not made of cells, and they are not considered to be fully alive. They have some characteristics of living organisms, but they lack the full set of characteristics of living organisms that biologists agree are required for something to be considered living. And we'll also talk about that in just a moment. So here is a size scale where you can see some common viruses and how they compare to other types of microorganisms. Here we have an E. coli bacterium, and here we have an elementary body of a chlamydium bacteria, which is one of the smallest types of bacteria in existence. And we can also see this huge human red blood cell here to compare as well. So among the smallest types of viruses are bacteriophages, and these are all the way down at 24 nanometers in size. We will also talk about bacteriophages later. Some human viruses that are particularly small are poliovirus, rhinovirus, and adenovirus. Larger viruses exist too. For example, the Ebola virus is a very large virus that is 970 nanometers in size. But as you can see, in general, viruses are so much smaller than even bacteria that you can fit many viruses inside of a single bacterium. And in fact, there are viruses that are dedicated to infecting bacteria called bacteriophages. And we'll talk about those as part of our lecture as well. There are viruses that have more recently been discovered, which are larger in size and more on the order of bacteria in terms of both their size and their genetic complexity. One group of these viruses is called pandoraviruses, and these viruses are thought to perhaps represent some sort of an evolutionary stepping stone between cellular life and viruses. Although much more study is needed to understand what the relationship is between pandoraviruses and cellular life. Now, let's go back to that point about why viruses aren't fully alive. Viruses cannot reproduce themselves or conduct metabolism independently without infecting a host. They are inert without a host cell to infect, and they are fully reliant on the enzymes and other molecular machinery found in host cells in order to generate new virions. Virions are viral particles, and so the ability to reproduce is one of those universal characteristics of living organisms that biologists acknowledge must be present in order for something to be considered alive. Viruses do reproduce. In fact, if you can see them reproducing in this electron microscopy image, each of these little dots is a virus that has infected this larger cell. So they can reproduce, but they can't do it without a cell, and therefore they are not a full-fledged form of life. So the bulk of this lecture is actually going to focus on the processes by which viruses conduct reproduction inside of host cells, because these are the processes by which viruses bring about disease. But before we can talk about their reproduction and their life cycle, we need to talk a little bit more about their structure. As we mentioned on the previous slide, the term that we use to describe a fully developed infectious viral particle is called a virion. As you can see, the structure of a virion can differ quite a bit from virus to virus. Here we see a variety of viruses from HIV, hepatitis B, Ebola, adenovirus, influenza, and bacteriophages. However, regardless of the type of virus and the disease that it causes, they all have the same basic structural components. They are composed of nucleic acids surrounded by a protein capsid, and sometimes they can also include an envelope and special viral enzymes, although these last two components are not found in all viruses, only in some of them. So let's take a look at each of these components one by one in a little bit more detail. Viruses are unlike any other types of biological entities because their genetic material exhibits a lot more diversity than what we see in the rest of the world. So we have learned always that DNA is the genetic material. But for viruses, their genetic material can either be DNA or RNA. We learned about RNA in our previous chapter, and we learned that it is an intermediary molecule that happens between transcription and translation in living cells such as our own. But in viruses, RNA can be the basic genetic material. Nucleic acids can also be single-stranded or double-stranded. We previously learned that RNA tends to be single-stranded and DNA double-stranded in cellular organisms. But in viruses, you can have single-stranded DNA or you can have double-stranded RNA. There's any possible combination of the two different types of nucleic acids with the two different varieties of strandedness. So this is another thing that makes them different. And the type of nucleic acid that they have and the strandedness of that nucleic acid bears on the reproductive process. Meaning, a virus that has a single-stranded RNA is going to reproduce differently than a virus that has double-stranded DNA, for example. And then finally, nucleic acids found in viruses can either be linear in nature or they can be circular. They can be composed of a single long strand of nucleic acid, or they can be composed of a continuous loop of nucleic acid. The structure of the nucleic acid and the type of nucleic acid is connected to the capsid shape that a virus has. So the capsid, as we said, is this protein coating or protein sheath that protects the viral genetic material. Here we can see two different shapes of capsids, and we'll talk more about these in just a moment. But the capsid is composed of repeating protein units called capsomeres. In this image right here, we can see very clearly the capsomeres, and then a capsomere has been highlighted right here. The shape depends on the type of nucleic acid. And these capsids, when they are barren, meaning without an envelope, they will be covered in protein spikes, which you can see in this figure down here. The spikes are highlighted here, and these protein spikes play an important role in the virus's ability to interact with host cells and perform the initial steps of the infection process. So there are a few different morphologies or capsid shapes that are recognized. The first is a helical virus. A helical virus has a capsid that is a long rod shape with a cylindrical capsule. The capsomeres are arranged into a helix, which is in response to a nucleic acid found internally that is also shaped like a helix. So the structure of the capsid is made to match the structure of the nucleic acid that is found inside it. An example of a helical shaped capsid virus is the Ebola virus, which you can see right here. Another example is the tobacco mosaic virus, which, although it's not a virus that's infectious for humans, it infects a variety of plants. It was the first virus ever to be recognized, the tobacco mosaic virus. And then there's polyhedral viruses. Polyhedral viruses have a capsid shape that is the three-dimensional shape of an icosahedron. An icosahedron is this 3D shape that has 20 equilateral triangular faces. And you can kind of see the structure of the icosahedron here in 3 dimensions, but when you're looking at polyhedral virus under the microscope, it often just looks like a regular hexagon because you can't discern the three-dimensionality of those 20 equilateral faces, because all you can see is this basic hexagon shape. The third category of capsid shape is called a complex structure. Complex viruses have a capsid shape that does not conform to either one of the two main capsid shapes. So for example, bacteriophages right here, they actually exhibit a combination of two capsid shapes in one. They have a helical shaped sheath or tail, and they have a polyhedral shaped head. Pox viruses over here clearly have a capsid shape that does not resemble either the helical or the polyhedral structure of a capsid. So in our first checkpoint, I want you to put that information to the test by looking at this electron microscope image of Marburg virus and tell me what capsid shape do we see here. So we've talked about capsid shape, we've talked about nucleic acids, and that brings us to a third component that is found in some viruses called the envelope. Regardless of the capsid shape, a virus may have something called an envelope over its surface. So an enveloped virus is one that either has a helical or polyhedral capsid that is enclosed in it. This additional layer of material on the outside of it is called an envelope. If a virus does not have an envelope, it is referred to as a naked virus. Both are possible. You can have a virus with an envelope, or you can have a virus that is naked. As you can see right here, there is a helical capsid inside of this larger envelope. So the shape of the envelope does not necessarily indicate to you what the shape of the capsid is inside. So here we have a helical capsid, and on the outside we have a spherical envelope. So the envelope, what is it? Well, it's a layer of lipids, proteins, and carbohydrates that may cover the viral capsid of some viruses, and it is similar in structure to the cell membrane in that it is a layer of phospholipid-like fats that has various other compounds embedded in it, similar to a fluid mosaic model. When an envelope is present, it will be covered in those protein spikes that we referred to previously, and the protein spikes, whether they are found on the surface of the capsid or the surface of the envelope, play a very important role in defining the virus's host range. And that's because the virus's ability to infect and get inside of a host cell depends on whether those spike proteins are compatible with receptors on the surface of the host cell. Every host cell has these protein receptors that are sticking off the surface embedded in the membrane, and viruses that are able to attach to those receptors and trick the cell into letting it inside will be able to initiate an infection. The component of the virus that attaches to those receptors is called the spike protein. If the virus is enveloped, then the spike proteins are the ones that are found on the surface of the envelope. If the virus is naked, then the spike proteins will be found on the surface of the capsid itself. So this compatibility between the spike proteins and the receptors on the host cell defines the host range, which is the range of species and or cell types that a given virus can infect based upon its attachment compatibility. For example, to give you some ideas of the restricted host ranges of different types of viruses, bacteriophages are a type of virus that can only infect bacteria. They are harmless to human and other animal cells. They only infect bacteria. The H1N1 influenza virus infects humans and pigs. In fact, in this figure right here, we can see a depiction of the host range of different strains of influenza virus depending upon their type. We can see H1N1 under humans and pigs, but there are also other varieties of influenza virus, like, for example, H13N9 that infects whales. So the range of influenza virus is limited by its protein spikes. There are also viruses that can infect plants. The cucumber mosaic virus affects over 1,200 different species of plants. However, in this lecture, we're not going to focus on viruses that infect plants. We are just going to focus on the viruses that infects humans. So now we've talked about nucleic acids, we've talked about capsids, and we've talked about envelopes. The fourth and final structural feature of viruses that we need to discuss before we can talk about their replication processes is special enzymes. Remember that, like envelopes, only some viruses feature special enzymes that are carried in their capsid alongside their genetic material. And these enzymes play important roles in the virus's ability to carry out infection. So, for example, bacteriophages, which are, you may recall, the types of viruses that infect bacteria, carry an enzyme called phage lysozyme, which is used to break the cell walls of their hosts so that they can allow their nucleic acids to enter the host cell. Another example of an enzyme is called protease. Protease is an enzyme that is carried and used by HIV. And HIV virus uses it to assemble new virion particles. So I have a second checkpoint for you. The Delta variant of COVID-19 is known to exhibit 3 mutations in its spike protein. It is at least 50% more transmissible than its precursor variant. So explain, based on what you now know, how mutations of this type can impact transmissibility of a virus. So now that we've finished covering the basics of the structure of viruses, we can start talking about the viral replication process. The mechanisms that viruses use to replicate can vary in slight ways, but they all follow the same basic series of steps. First comes attachment. Attachment is where the virus adheres to receptors on the host cell using its protein spikes. After attachment comes penetration. If the spikes are compatible with the receptors on the host cell, then the virus will penetrate the host cell wall and or membrane depending on the type of cell that it's infecting. And then comes biosynthesis. The virus hijacks the host cell in this step in order to force the host cell to redirect its attention away from its normal metabolism and instead to focus on making more parts of new viruses, including proteins for the capsid and copies of the nucleic acid material. Then comes maturation. When biosynthesis takes place, it makes the pieces of the new viruses, but those pieces need to be assembled. For example, the capsomeres have to be constructed into the new capsid, and so the newly made parts are assembled into full viruses in the step called maturation. The virus becomes mature. And lastly comes release. During release, the new viruses are released from the host cell in order to go on and infect other cells and propagate the viral replication. Now, as I said, the replication cycle can exhibit variations even though it has the same basic series of steps. And so we are going to look at a few different types of viruses that vary in their nucleic acid and their host range. We're going to take a look at bacteriophages and animal viruses. And within the category of animal viruses, we are going to look at DNA viruses, RNA viruses, and retroviruses. So let's start with bacteriophages. So bacteriophages have bacteria as their hosts, and as we've seen, they have this crazy alien-like complex morphology where they have a polyhedral head, a helical tail, also called the sheath, and they have these tail fibers that represent their spikes that are used to attach to the surface of their bacterial cell hosts. They are non-enveloped, meaning they are naked. Their capsid does not have an envelope covering its surface, and their genetic material is composed of double-stranded DNA. Bacteriophages, depending upon the variants, can exhibit two different life cycles. Virulent phages are bacteriophages that perform something called the lytic cycle, and temperate phages perform something called the lysogenic cycle. Both of these cycles results in the production of new bacteriophages, but one of them is more immediately violent for the host cell than the other, and that is the one performed by virulent phages. Temperate phages are much more sneaky and less immediately violent, as we will see as we go over these two different cycles in just a moment. So let's start by looking at the lytic cycle, the cycle performed by virulent phages. In the first step of attachment, those tail fibers that we saw on the previous slide will attach to the receptors on the cell wall of the bacterial cell hosts that has been targeted by the virus. After that comes penetration, wherein the viral enzyme phage lysozyme breaks down part of the cell wall, and then the sheath of the capsid contracts, kind of like a hypodermic needle, and it injects the DNA into the host cell. The capsid remains external to the host cell, so the capsid does not enter, rather it simply injects its DNA into the inside of the host cell. So after this step, all we have is viral DNA inside of the host cell. Next comes biosynthesis. That little piece of DNA will shut down the normal transcription and translation of the host cell, and it will hijack the enzymes, nucleotides, ribosomes, and amino acids found in the cell and put them to work making new viral DNA and proteins, the pieces of new viruses. During maturation, those pieces, viral DNA and proteins, will spontaneously assemble into new, fully formed virions. And finally, during release, after a certain number and concentration of new viruses have been produced, the host cell will lyse, or in other words, burst, which of course kills the cell and releases all of those new virions so that they can go off and find new hosts. So as we can see, the lytic cycle ends in the death of the host cell pretty quickly. And the namesake of the lytic cycle is the final and ultimate blow to the cell, the lysis. The lytic cycle is named for the lysis that happens at the end of the cycle. We can contrast the lytic cycle to the lysogenic cycle in that the lysogenic cycle, whereas the lytic cycle we can think of as like a hostile takeover of the cell, we can think about the lysogenic cycle as being more of a covert operation. During the lysogenic cycle, attachment and penetration take place just like they do in the lytic cycle. But that's where things start to change, because after the viral DNA has entered the host cell, something called integration takes place. Integration is where the viral DNA will actually merge with and become part of the bacterial cell's chromosome. So as we can see, this little fragment of green DNA in here, that represents the viral DNA that has merged with the bacterial DNA. So in this sense, phages that perform the lysogenic cycle are sneaky. They will sneak their DNA into the bacterial chromosome, and in doing so, they create what's called the prophage. The prophage, literally meaning prephage or previrus, can exist inside of a strain of bacteria for literally generations indefinitely, because every time this bacterium undergoes binary fission and every time it clones itself, it is going to unwittingly also propagate this piece of viral DNA along with the rest of its chromosomes. Now, this can go on forever, and the viral DNA can be propagated in this manner indefinitely. But sometimes a phenomenon called induction can occur. Induction is where the bacterial cell realizes that it has been had, and the piece of viral DNA is kicked out of its chromosome, shown right here. Induction can be triggered by environmental forces like stress, like exposure to UV light or exposure to certain chemicals. But regardless of the reason for induction, once induction happens, the virus will feed into the lytic cycle and initiate the hostile takeover. Once this piece of DNA is kicked out, the virus immediately jumps into biosynthesis mode and starts taking over the host cell to generate more virions. The virions mature and ultimately are released through the lysis of the host cell. So yes, the lysogenic cycle can end in destruction of the host cell, but it does not necessarily need to do so because propagation can go on literally indefinitely. Now, when a bacterial cell is infected with a temperate phage that carries out the lysogenic cycle, there are a few consequences that occur for that bacterial cell which are not relevant to the lytic cycle. One of those consequences is that the bacterium becomes immune to reinfection with the same bacteriophage. Another one is a phenomenon called phage conversion. Phage conversion is what happens when the bacterial cell traits are changed by the presence of the prophage in its genome. So, for example, there are many examples of bacteria that are normally harmless to humans, but when infected with a lysogenic phage, they can be transformed and start generating toxins that suddenly make them more virulent and harmful to humans. So phage conversion is something that can actually take a bacterial cell and through infection of that bacterial cell with a virus, turns it into a more harmful version of itself by altering its traits. A third phenomenon that can occur is something called specialized transduction. Specialized transduction describes the manner in which fragments of the bacterial cell's chromosome or its genome can be cut out along with the prophage when induction occurs. And when this happens, those fragments can be packaged into a new viral particle, which will then transfer them to a new host cell after it is released. So let me show you a clearer picture of how this works. We know that during induction, This little piece of viral DNA will get cut out or excised from the bacterial chromosome, but sometimes the bacterium is sloppy about how it goes about doing this. Instead of cutting right along the edges of the viral DNA, it will chop somewhere off center from the viral DNA and capture a little bit of its own DNA alongside the prophage. That means that the viral DNA will now be composed of a little piece of the bacterial DNA along with it. And when this viral DNA is stuffed inside of a capsid, and that capsid is released from the original host cell as it bursts, the virus that goes on to infect new hosts will bring with it a piece of the genetic material from the previous bacteria. And so this can actually allow new bacteria to receive traits from previous bacteria by being carried accidentally through viruses. So in this checkpoint, I want you to have a look at the bacteriophage podovirus here. Podovirus does not integrate its DNA into the host cell genome. So tell me, which viral life cycle does it exhibit? And would you describe podovirus as virulent or temperate? One last thing I want to mention while we're on the topic of bacteriophages is that although we have talked about how they are harmful for bacterial cells and how they can make bacterial cells more harmful to humans through phage conversion, we are also learning how they may be harnessed in order to fight bacterial infection. One of the things that I mentioned to you at the end of our very first chapter's lecture is how bacteriophages are being deployed to fight resistant infections, particularly occurring in people who are genetically afflicted by cystic fibrosis. There was a video that I referred you guys to in the first lecture which walked through the process by which these bacteriophages can actually be packaged into an inhaler that someone might use for asthma, and then breathed in for a person who suffers from cystic fibrosis to allow those bacteriophages to go to work on the infection taking place in their lungs. So it's been rather successful and a very interesting new route to pursue as an alternative to traditional antibiotics, especially as bacteria become more and more resistant to antibiotics that we've used for hundreds of years. So this completes our discussion on the topic of bacteriophages. And now it's time to move on to our discussion on animal viruses. Within the topic of animal viruses, we have three subtopics that we're going to be covering, or three different types of animal viruses. But this is the point at which, if you want to take a break from the lecture and step away for a while, this is an ideal place to do it because we're in between topics right now. And so we should be at approximately at the halfway point now. So feel free to take a break and then come back and resume the rest of the lecture focusing on animal viruses. So now we're going to move on to discussing animal viruses. The hosts of animal viruses are animals, which includes ourselves. Although the species and cell type varies, the host range is different for each animal virus. They exhibit a wide variety of capsid shapes. They can be polyhedral, helical, or complex. And they exhibit multiple nucleic acid types. So we're going to be talking about RNA viruses, DNA viruses, and retroviruses individually as we talk about animal viruses at large. Before we start talking about the different subcategories of animal viruses, we are going to take a look at a broad overview comparison of the differences between bacteriophages that we just talked about and animal viruses in terms of the stages of their replicative life cycle. So with bacteriophages, during the attachment stage, we saw that the tail fibers adhere to receptors on the cell wall, but in animal viruses, the protein spikes on either the envelope or the capsid are what adhere to receptors on the cell membrane. Remember that animal cells do not have a cell wall, they only have a cell membrane, and so this is where the receptors for the viruses are located. In penetration, we saw that bacteriophages would contract and inject their DNA genome into the host cell from their capsid. The capsid did not enter the host cell. However, with animal viruses, the entire capsid will enter the host cell via one of two processes, either endocytosis or fusion. The difference between endocytosis and fusion is that fusion describes the process of entry by an envelope virus, where this layer of material that we call the envelope fuses with the cell membrane and dumps the capsid inside. Non-envelope virus cannot perform fusion because they don't have an envelope with which they can fuse. Instead, non-envelope viruses perform endocytosis, which is the process by which the cell membrane will create a vesicle or a pocket of membrane material that brings the virus inside. Envelope viruses can either perform fusion or endocytosis, but non-envelope viruses can only perform endocytosis. So getting back to our comparison, because of the fact that the entire capsid enters the cell, which we did not see in bacteriophages, there is an additional step in the life cycle of animal viruses that is not present in bacteriophages, and that step is called uncoating. Uncoating is where the capsid releases the genetic material into the cytoplasm. Biosynthesis in bacteriophages involve taking the DNA, replicating it, and transcribing and translating it to generate viral proteins. For animal viruses, because they have such a wide variety of different nucleic acid types, there are a variety of different biosynthesis mechanisms by which the viruses replicate. So that's why we have to break down our virus discussion into DNA, RNA, and retroviruses, because this is really where the biggest differences take place. How does the virus initiate biosynthesis of new viral parts? It varies depending on type. Maturation in bacteriophages took place spontaneously. Remember that maturation was the assembly of the viral parts that were produced during biosynthesis. In animal viruses, that maturation process may require the assistance of special enzymes that put those parts together. And finally, in release, we saw that bacteriophages would kill their host cells via lysis at the end of their life cycle, whereas animal viruses may not kill their host cell. In some cases they do, but in other cases they can be released through a process called budding. Budding is like the opposite of endocytosis, whereas in endocytosis a vesicle is created that allows the viral particle to enter. In budding, the membrane material creates a pocket which eventually transforms into the envelope of the virus, and in this manner the capsid of the virus is able to leave and take with it part of the cell membrane that transforms into its envelope. This of course does not kill the host cell and allows it to continue living and turn it basically into a virus-making factory that has been hijacked by the virus and keeps budding off additional viruses. So our next checkpoint is related to this cartoon that I found, which depicts one of the two forms of animal virus penetration. We can see that there is a virus here on the outside. We can see that other viruses are merging, and then later on they are breaking open and releasing the contents of their capsid. So which animal virus penetration mechanism do we see happening in this cartoon? Is it endocytosis or fusion? So out of the three different subtypes of animal viruses that we are looking at, we are going to start with DNA viruses. DNA viruses are ones that have DNA as their genetic material, and for these viruses, there is a relatively straightforward process for them to conduct their replicative life cycle in a host. After they attach to penetrate and uncoat within the host cell, the viral DNA will enter the nucleus being the location where the cell's normal DNA is found, and it will be transcribed and translated into new viral proteins. It will also be used as a template for replicating new pieces of viral DNA. This, of course, is the biosynthesis. Then the viral proteins and DNA will be assembled and mature into new fully developed virions and will be released typically through budding. So there are a variety of different DNA virus families that categorize different groups of viruses based upon their relatedness. Typically, these groups of viruses are not only genetically related, but they cause similar diseases. So, Adenoviridae is a family of double-stranded DNA virus that causes respiratory infections such as the common cold. Poxviridae is a family of double-stranded DNA viruses that causes disease associated with skin lesions, including smallpox and cowpox. Herpesviridae, also double-stranded, causes diseases associated with sores and rashes, including cold sores, chickenpox, mononucleosis, and roseola. Papillomaviridae is a double-stranded virus that causes diseases that are associated with causes of cancer, especially cervical cancer, as well as warts. And Parvoviridae is a single-stranded DNA virus that also causes diseases that are associated with rashes, including fifth disease. Fifth disease is an infection mostly found in children. It leads to common cold-like symptoms followed by a bright red rash on the face and sometimes on the body. So while DNA viruses are straightforward, RNA viruses are a little bit more complex. And this is mainly because the biosynthesis of an RNA virus varies depending upon whether the RNA virus is double-stranded, single-stranded plus, or single-stranded minus. And we'll talk about what that means in a second. But before we do, I'm going to point out some important RNA virus families that cause diseases in humans. The Picornaviridae is a single-stranded positive RNA virus family that includes other causes of the common cold and the virus poliomyelitis. Togaviridae is also single-stranded positive. It includes viruses that cause encephalitis and rubella. Retroviridae is a single-stranded positive virus family that includes the cause of AIDS. Filoviridae is a single-stranded minus virus family that includes causes of hemorrhagic fevers such as Ebola. Orthomyxoviridae is a single-stranded minus family that includes influenza viruses. Rheoviridae is a double-stranded that includes gastroenteritis. Now, I don't want you guys to know whether or not these are single-stranded plus or single-stranded minus or double-stranded. It's more important that you connect the name of the family to the types of diseases that it causes. So now let's talk about what it means to be single-stranded plus versus single-stranded minus, or double-stranded for that matter, because this is something that might be revolutionary to us at this point. RNA can be double-stranded in viruses. Up until now, you may have learned that RNA is a single-stranded molecule, but in viruses, it can be double-stranded. And when it is double-stranded, we have a special designation for one strand versus the other in order to keep them straight from each other. The positive strand is designated with a plus, and the negative strand is designated with a minus. So some RNA viruses have both of these strands stuck together in a double-stranded configuration, but it's also possible for viruses to have either one or the other. Some RNA viruses just have a negative RNA strand. Others simply have a positive RNA strand. And as we said, this has bearing on how the biosynthesis of new viruses takes place. There are a couple of rules that, if you know these rules, you should be able to figure out how an RNA virus replicates based upon what type of strand it has. So keep in mind that when new virions of RNA viruses are being made, there are two main ingredients that the cell needs to produce to generate these viruses. They need to make more RNA pieces, and they need to make more proteins to create the capsid that encases them. Plus strand RNA can be used as a template to make minus strand RNA. And likewise, minus strand RNA can be used as a template to make plus strand RNA. The way that it works is it is extremely similar to the process for replicating DNA that we saw in our last lecture, where one strand is used as a template and free nucleotides are then matched up to that strand by an enzyme to create a new strand that has the opposite sequence. So this can happen not only with DNA, but it can also happen with RNA. Plus strands can make minus strands, and minus strands can make more plus strands. However, only plus strands are able to be read by ribosomes and therefore used to make protein. Protein is essential for making the capsid, and so we need plus strands in order for the ribosomes to read them and make more protein, even if it's a minus stranded virus. So let's take a look at a comparison of how positive and negative stranded RNA viruses replicate based on these rules that we just went over. If we're looking at a single-stranded positive RNA virus, we know that to generate new virions, the cell will need to create more plus strands and it will need to create protein capsids, so that is the goal. However, we can't create plus strands from plus strands. We can, however, generate proteins from these plus strands because they can be recognized by the ribosomes. And so the plus strands will be read by the ribosomes to generate protein. And then they will also be read and used to replicate minus strands because we can make minus strands from plus strands. But then those minus strands will have to serve as a template for making more plus strands. We can combine the plus strands, stuff them in a capsid made from proteins, and now we have what we need to make more single-stranded positive RNA viruses. The process looks different for single-stranded minus RNA viruses. These guys need to make more minus strands, and they need to make more protein capsids. The minus strands cannot be used as a template for the ribosome, but it can be used as a template for making more plus strands. So the negative strand will be used to generate positive strands. The positive strands can then be read by the ribosomes to make proteins, and they can be read to replicate their complementary negative strands. You take those negative strands, you put them in a capsid, and now you have more identical single-stranded negative RNA viruses. The last type of RNA virus is the double-stranded RNA virus. Of course, this guy needs more double strands, and it needs more protein capsids. So what will happen once inside the cell is the double-stranded RNA will be unwound, the minus side will be read and used to replicate more plus strands, which are read and used to make more protein capsids by the ribosome, as well as generate complementary negative strands, which, when put together, give you more double strands. Put these inside of a capsid, and you have more identical double-stranded viruses. So as you can see, the mechanism differs depending upon what type of nucleic acid there is. However, if you simply remember those three rules, that minus strands make more plus strands make more minus strands, and only plus strands can be read by the ribosome to make protein, then the sequence of steps that occur for each different type of RNA virus should be pretty logical. Our third and final category of animal viruses to discuss are the retroviruses. So far, we've talked about DNA viruses, we've talked about RNA viruses, and now we're going to talk about retroviruses, which are actually a special subcategory of RNA viruses. The thing that sets retroviruses apart from other types of RNA viruses is that they possess a special enzyme called reverse transcriptase, which takes their RNA genome and converts it to DNA. If you think about the name of reverse transcriptase, we previously learned that the process of transcription occurs in all of our cells, and it is the process of taking our DNA and making a copy of it in the form of RNA. So therefore, reverse transcriptase is an enzyme that performs the reverse process of transcription. If transcription converts DNA to RNA, reverse transcriptase is the enzyme that turns RNA into DNA. The thing that makes these viruses so virulent is that once their RNA has been turned into DNA, that DNA will integrate into the host cell chromosome and become part of your DNA, much like a temperate bacteriophage is able to do in the lysogenic cycle. The most well-studied and well-characterized retrovirus is human immunodeficiency virus, or HIV. And one of the reasons why HIV is so difficult to cure and treat is because it is a retrovirus where the viral DNA is turned into DNA and then it becomes part of an infected person's DNA. On the screen here, you can see a depiction of the life cycle of a retrovirus. So let's walk through the steps. If this is the HIV retrovirus, the first step is to attach to the receptors on the host cell. After that, the virus penetrates via fusion and uncoats its capsid to release its RNA and its reverse transcriptase enzymes into the cell. That RNA will be picked up by the reverse transcriptase enzyme and converted into DNA. After that, the DNA enters the nucleus, and another viral enzyme called integrase will cause that DNA to become integrated into the human chromosome. Next, the human chromosome will undergo regular biosynthesis, it will be replicated, and the genes will be transcribed and translated to create new viral proteins. The pieces of new HIV virus will be assembled through maturation with the assistance of another enzyme called protease. And lastly, new HIV virions will be released from the host cell through budding. So in fact, this viral production process does not immediately kill the cells that it infects, which are cells of the human immune system. However, over a long period of time, with a long duration of infection, eventually these cells, which have been transformed into HIV-making factories, will start to die. Scientists aren't entirely clear why, but we know that it's not through the release of the virus that they die. The viruses are released through budding. However, over time, the immune system cells do start to decline, and that's when a person begins to experience the acquired immunodeficiency syndrome, or AIDS. That results from a long-term HIV infection. And I should say that not every long-term HIV infection progresses to AIDS, because as we will talk about later, there are actually a wide variety of antiretroviral drugs out there that are able to treat HIV infections very effectively and keep the disease from progressing to the point where it is considered AIDS. Scientists have developed such effective drugs for treating HIV infections that the average life expectancy of a person who has HIV infection is actually not that far off from the average life expectancy of someone who does not. But this is something we can talk more about later. For now, we are going to talk a little bit about treating viral infections in general. There are antiviral drugs that have been developed for specific types of viruses that inhibit a specific stage in their replication cycle. One that you may have heard of is Tamiflu. Tamiflu is a drug that is used to treat influenza virus infections. The way that Tamiflu works is by inhibiting the release stage of virions. It interferes with the enzymes on the flu virus that are involved in budding. So Tamiflu doesn't actually prevent the virus from entering your cells. It doesn't actually prevent the virus from undergoing biosynthesis and maturation. What it does is it stops new viruses from exiting already infected cells, and in doing so, it prevents the infection from further spreading, makes a person less contagious, and lessens the severity of the symptoms and duration of the disease. Zidovudine is another antiviral drug. This one is used to treat HIV. It interferes with the conversion of the viral RNA to DNA by inhibiting the reverse transcriptase enzyme that is part of the HIV virus. Here's another drug that you may or may not have heard of, which is also used to treat the flu. The way that amantadine works is by inhibiting a viral protein on the flu called M2. M2 allows the viral capsid to release its content into the cell. So tell me, which stage of the viral life cycle of the flu is targeted and inhibited by amantadine? And this checkpoint will require you to think back to one of our previous lectures back in Unit 1. Acyclovir is an antiviral drug that is used to treat chickenpox and herpes infections. The way that it works is by binding to the active site of the viral DNA polymerase enzyme that is responsible for synthesizing new genetic material for future viruses. So I want you to tell me, what type of enzyme inhibitor is acyclovir? Is it a competitive or non-competitive enzyme inhibitor? So this completes our discussion on viruses. And now we're going to circle back to this very first slide where we took a look at not just viruses, but all the acellular pathogens that have been characterized by microbiologists. We are also going to briefly look about viroids, virusoids, and prions here in this last part of this lecture. Viroids and virusoids are unique types of pathogens that are not composed of a combination of protein and genetic material, but rather just pure genetic material. Both of them are composed of RNA without a protein capsid. Viroids are composed of a short strand of circular RNA, so a little RNA loop, while virusoids are composed of a single-stranded piece of RNA. The difference between the two is that viroids are able to self-replicate after infecting their host cell, which is usually a plant. Virusoids are only able to replicate inside of host cells that have also already been co-infected with a helper virus. And lastly, we have prions. Whereas viroids and virusoids are the genetic material without the protein component of a virus, prions are the protein without any genetic material. These are pathogenic proteins capable of corrupting other proteins and leading to the formation of plaques. A disease-causing prion protein, typically abbreviated PR, capital P, with a little subscript which indicates what type of disease we're talking about, will approach other proteins and it will essentially change their shape and transform them into pathogenic proteins like themselves. And as the quantity of these proteins build up, it forms plaques in the brain that inhibit neurological function. So all known prion diseases are neurodegenerative diseases of the brain. They are often called spongiform encephalopathies because the buildup of these plaques leads the brain to take on a spongy texture. So as you can see on the slides on the right, you can see there is normal brain tissue on the top, and below you can see these sponge-like lesions that have appeared in the brain tissue of a patient who has Creutzfeldt-Jakob disease, which is one type of prion infection. The other wild and crazy thing about prion diseases is that they are both inheritable, meaning that a person can be genetically predisposed to a prion disease and pass that genetic predisposition down to their children, and they can also be transmitted. This is the only disease where it can be a heritable disease that can also be contagious to another person. And that's because these prion proteins can be transmitted from a person who has a genetic prion disease to someone who does not. For example, pathogenic prion proteins have been documented transmitting from someone who has a prion disease to someone who does not through a corneal transplant. They have also been documented passing from a patient to a surgeon when accidentally cut with a scalpel. Some examples of known prion diseases include Kuro, bovine spongiform encephalopathy or mad cow disease, Creutzfeldt-Jakob disease, and fatal familial insomnia. Here on this slide, we can see a brain that has been affected with Creutzfeldt-Jakob disease and developed that spongy texture versus a control brain. Mad cow disease is one that you may have heard of before. There was an outbreak of mad cow disease in the late 80s and early 90s in the UK. And these proteins could be transferred to humans that people who lived in the UK during a certain period of time are prohibited from donating organs or giving blood in case they are afflicted unknowingly by a prion disease. Kuru was the first ever recognized human prion disease, and this prion disease was observed in the Foray tribe in Papua New Guinea, which is an island off the coast of Australia. And it was discovered that what had happened is one person had likely developed a genetic mutation that led to the prion disease. And because part of the cultural and spiritual tradition of this tribe was to cannibalize the brain matter of their dead loved ones, the disease then spread throughout the community over the course of decades. Prion diseases have a very long incubation period, and so it was only very recently, in the early 2000s, that this community, this tribe, was declared finally free of this prion disease that had existed for so long since the 1950s. Luckily, prion diseases are extremely rare. However, they are also incurable. So there is research going on right now on how to possibly treat and cure prion diseases. Although as of right now, there is no cure. Now, in this very last part of this lecture, we are actually going to take a step back and look at a section of Chapter 11 that we didn't cover in our Chapter 11 lecture. And the reason why is because we couldn't really wrap our heads around this until after we talked about viruses. Chapter 11, Section 6 is all about how prokaryotes acquire genetic diversity. Genetic diversity is important because it provides a species with robustness. Populations of organisms are better able to survive when their members exhibit a wide variety of traits. This is not an issue in eukaryotic organisms because eukaryotic organisms often reproduce sexually. Sexual reproduction takes the genetic information of two partners and mixes it and combines it into new ways, which generates diverse outcomes. However, prokaryotes, as we've learned, reproduce asexually by cloning themselves through the process known as binary fission. And so, they have two alternative sources of genetic diversity that do not come from sexual reproduction. And those sources are mutations or random errors in the genetic code, as well as methods of horizontal gene transfer. We've already talked about mutations, so now we're going to talk about horizontal gene transfer. To understand horizontal gene transfer, it's important to first understand what vertical gene transfer is. Vertical gene transfer is the passage of genes from an organism to its offspring. It's called vertical gene transfer because it involves passage of genes down the generations, so in a vertical direction. Horizontal gene transfer, on the other hand, refers to the passage of genes between organisms of the same generation. Humans cannot do this. Animals can't do this at all. Prokaryotes are pretty much the only organisms that can do this. And there are 3 different methods by which they can transfer their genes horizontally. One is conjugation, another is transformation, and the third is transduction. Conjugation is something that we've talked about before. It's the process of transferring plasmids through direct contact between cells. When we're talking about a gram-negative species of bacteria, conjugation takes place through a pilus or pili. There are special conjugated plasmids that encode the genes which give a bacterium the ability to create a pilus and then transfer that plasmid through the pilus to another bacterium. Gram-positive bacteria have a slightly different approach. These cells produce a sticky surface called a mating bridge, and it is through that sticky mating bridge that the DNA plasmids are able to transfer. Here we can see a diagram that shows conjugation in action in a higher level of detail. The plasmids that undergo conjugation are referred to as F plasmids, where F stands for fertility. They're called fertility plasmids because the fertility is referring to the way that the plasmid is able to transfer from one organism to another and therefore gives genes to another organism. What will happen is the F plasmid will allow the donor cell to create a pilus or a mating bridge, and then that plasmid will be replicated and a second copy of it will be sent through the pilus or the mating bridge so that the other cell, which was previously referred to as an F negative cell, now becomes an F positive cell along with the original donor. Conjugation is an important process for not only bacteria but also for humans, because conjugation has been documented to transfer important virulence factors between bacteria. The gene for verotoxin, which is the cause of the disease dysentery, has been seen transferred from the organism that causes this normally, Shigella dysenteriae, to regular old harmless E. coli, suddenly giving E. coli the ability to generate this toxin. Genes for the tetanospasmin toxin, which is involved in the cause of tetanus, have also been transferred via conjugation from Clostridium tetani to other non-pathogenic bacteria. Antibiotic resistance has also been documented as being transferred through conjugation between bacteria. So genes for methicillin resistance in MRSA, which stands for methicillin-resistant Staphylococcus aureus, have been transferred from Staphylococcus aureus to other bacteria, as well as genes for vancomycin-resistant Staphylococcus aureus, or VRSA. So conjugation, while it can be a great thing for the survival of bacteria in order to increase their diversity and provide them with new genes, it can be a bad thing for humans because it allows bacteria to share these genes that increase their ability to cause disease and survive under the presence of our antibiotics. Transformation is the second part or the second type of horizontal gene transfer. And transformation is the process of naked DNA being passed between cells. Naked DNA means that DNA that is floating around in solution, not contained within a cell. And there can be many reasons why naked DNA is present, including cell death. When cells die and when they lyse, their DNA is released into the environment. and then that DNA can be picked up by other cells. It can be brought inside, and then through a process called genetic recombination, it can actually merge with and become part of the new cell's chromosomes. And so it's really strange, but it's like taking up the genes of one of these cells' dead friends and then making it a part of its genes. And lastly, the third process called transduction is one that we mentioned previously. Transduction is the process of transferring DNA from one cell to another through a bacteriophage, or in other words, just a phage, as they are called. We saw that temperate bacteriophages can perform the lysogenic cycle, integrating their DNA into the host cell chromosome, and then accidentally picking up pieces of the host cell's DNA before releasing new phages that go on to infect other cells and inadvertently deliver pieces of the previous cell's genetic information, which can become incorporated into new cells. So this phenomenon of transduction is the main reason why we had to wait to talk about section 11.6 until after we have already talked about viruses, because it's difficult to wrap your head around how transduction works until after you know how viruses complete their replicative life cycle. We have one final checkpoint here to look at. Here you see a Gram stain, and what I want you to tell me is, which of the two organisms would you expect to form a pilus in order to perform conjugation? Would you expect the cocci or the bacilli to form a pilus? Once you've finished with this one, we are now finished with chapter 6.